To utilize lactose as an energy source, E. coli must produce the enzymes necessary for its metabolism. The genes for these enzymes are adjacent to one another on the bacterial chromosome and are transcribed together. A group of bacterial genes that are transcribed together is called an operon. At one end of the Lac Operon is a set of structural genes, Lac-Z, Lac-Y, and Lac-A, that encode the enzymes necessary for metabolizing lactose. These three genes have a common promoter. The regulator gene, Lac-I, is located upstream of the structural genes. The Lac-I gene encodes a repressor protein and is controlled by its own promoter. This repressor protein may bind to a region of the operon called the operator. The operator overlaps the 3' end of the promoter and the 5' end of the Lac-Z gene. The Lac Operon is inducible, that is, the structural genes are expressed only in the presence of lactose. Active repressor proteins are encoded by the regulator gene. In the absence of lactose, these repressors bind to the operator and prevent RNA polymerase from initiating transcription. When lactose is present, it is readily converted into allolactose, which may then come in contact with the repressor protein. Allolactose binds to the repressor protein and causes a conformational change that prevents the repressor from binding to the operator region of the DNA. With the operator site open, RNA polymerase is able to bind to the operon promoter and transcribe the structural genes. The resulting mRNA is then translated, leading to the production of the three enzymes, beta-galactosidase, permease, and transacetylase, encoded by Lac-Z, Lac-Y, and Lac-A, respectively. Lactose is a carbohydrate found in milk. It can be metabolized by E. coli bacteria that reside in the mammalian gut. Because lactose is too large to diffuse across the bacterial membrane, it must be actively transported into the cell by permease, encoded by the Lac-Y gene of the Lac Operon. To utilize lactose as an energy source, E. coli bacteria use the enzyme beta-galactosidase, encoded by the Lac-Z gene of the Lac Operon, to break lactose down into glucose and galactose. The function in lactose metabolism of the third enzyme, transacetylase, also produced by the Lac Operon, is not yet known. Partial diploids have been used to help define the roles of the different components of the Lac Operon. In this example, the bacterial chromosome has a wild-type version of Lac-I, Lac-O, Lac-Z, Lac-Y, and Lac-A. Mutated versions of these genes are then inserted on a plasmid. Let's see what happens when the plasmid contains a mutated Lac-I gene and a wild-type version of everything else. The wild-type Lac-I gene on the chromosome codes for a normal repressor, while the mutated Lac-I gene codes for a non-functional repressor. This non-functional repressor cannot bind to the operator regions of either operon, even in the absence of lactose. However, the active repressors are able to bind to operator regions of either operon where they prohibit the binding of RNA polymerase. Because RNA polymerase cannot bind, the structural genes are not expressed. In the presence of lactose, and thus allolactose molecules bind to the active repressor proteins. As a result of this allosteric inhibition, the repressor proteins can no longer bind to the operator regions. When the operator regions are open, RNA polymerase can bind and transcribe the structural genes. The transcribed mRNAs are translated into the enzymes needed for lactose metabolism. From this partial diploid, it's clear that the Lac-I plus gene is dominant over a Lac-I minus gene, and a single copy of the normal gene is sufficient to bring about normal regulation of enzyme production. What about a case in which the Lac-I mutation results in the expression of a superrepressor? The superrepressors lack binding sites for allolactose. However, like the normal repressors, the superrepressors are transacting and can bind to either operator region in the absence of lactose. With the repressor proteins bound to the operator regions, RNA polymerase is unable to bind, and the structural genes are not expressed. In the presence of lactose and thus allolactose molecules bind to the wild-type repressors. The allosterically inhibited active repressors can no longer bind to the operator regions. Allolactose cannot bind to the superrepressors, so these repressors are still available to bind to the operators of both operons. With the superrepressors bound, RNA polymerase cannot initiate the transcription of the operons. The structural genes are not expressed even in the presence of lactose. In the next example, the plasmid version of the LAC-I gene is normal, but the operator region is mutated. In the absence of lactose, repressor proteins from either LAC-I gene fail to bind to the mutated operator, but succeed in binding to the normal operator. Repressor bound to the lower operon blocks RNA polymerase from binding. However, no repressor is bound to the mutant operator to impede the polymerase. Thus, RNA polymerase transcribes the structural genes of the upper operon, and the mRNAs are then translated into the lactose metabolizing enzymes, even in the absence of lactose. In the presence of lactose and thus allolactose molecules bind to the active repressor proteins. As a result of this allosteric inhibition, the repressor proteins can no longer bind to the wild-type operator. Now both operons are available for transcription. RNA polymerase transcribes the structural genes. The mRNAs are then translated into the enzymes necessary for metabolizing lactose. This partial diploid displays the phenotype of the LAC-OC mutation, which is the constitutive expression of the LAC structural genes in the presence or absence of lactose. Thus, LAC-OC is dominant over LAC-O+. What happens if we combine a LAC-I superrepressor with a LAC-OC mutation? In this example, both normal repressors and superrepressors are made, but neither type can bind to the mutant operator. Repressor bound to the lower operon blocks RNA polymerase from binding. However, no repressor is bound to the mutant operator to impede the polymerase. Thus, RNA polymerase transcribes the structural genes of the upper operon, and the mRNAs are then translated into the lactose metabolizing enzymes, even in the absence of lactose. In the presence of lactose and thus allolactose molecules are unable to bind to the superrepressors. Allolactose can allosterically inhibit the wild-type repressors so that they can no longer bind to the wild-type operator. The superrepressors are still available to bind to the wild-type operator and inhibit transcription. Because the superrepressor cannot bind to the mutant operator, RNA polymerase is free to transcribe the upper operon. The resulting mRNAs are then translated into the enzymes necessary for metabolizing lactose.